Thank you for joining this product demo on Benchling's COVID-19 Testing Initiative, a clinical data management use case. Before we get started, I'd like to share a quick snapshot of Benchling as a company. Benchling was founded out of MIT back in 2012 by a team of scientists and engineers who were frustrated with the poor quality of software they had available to support their science. They began by building software tools for scientists to support biology research, but quickly realized the entire life science R&D industry needed to transform. Since then, Benchling has created a range of solutions for life sciences, all built on a common platform. Our solution closely followed the needs of our customers, and today we are fortunate to work with over 400 innovative life science companies, including many of the world's largest enterprises. These companies are making amazing discoveries and innovations, and our team is committed and well-equipped to provide the best possible software to support them. The Benchling team is committed to our mission to accelerate the life sciences for the betterment of humanity. The biopharma industry has evolved tremendously over the past decade, and we're proud to work with hundreds of leading companies that have helped develop new advances in medicine and discover new therapeutic modalities. The output of the life sciences industry has been steadily increasing. As major drugs have gone through loss of exclusivity, companies of all sizes have been racing to fill pipelines with the next generation of therapeutics. One way companies have helped drive this acceleration is by increasing their degree of specialization. There is an ever-increasing number of research and drug modalities being pursued right now that help treat disease in new, more targeted ways. The therapeutics landscape used to be dominated by small molecules and monoclonal antibodies just a few years ago, but it is now much more evenly distributed across newer modalities like adoptive cell therapies, antisense oligonucleotides, and more, but also an emerging category of hybrid modalities like antibody-drug conjugates that blur the lines between small and large molecule research. This requires highly specialized teams to bring these innovations to market, and most of our customers have organizational structures that reflect this. But with each of these teams focused in highly specialized areas, and each using the next generation of tools available to them, like sequencing, lab automation, and proteomics, there is an explosion in the amount of data being created. The challenge is not just about the volume of data, it's about the organization and structure to this data. In many companies, most of this data remains siloed and not analyzed in the broader context of their programs. While the changes in the industry have taken place steadily over the past decade, we see a much more intense pressure being placed on R&D organizations. This requires an entirely new R&D paradigm to keep up with industry demand. Modern life sciences R&D requires a new way of working that reflects both the rapidly changing scientific landscape as well as the market pressures companies face. To stay competitive in this industry, companies are focused on building scientific platforms that look beyond individual discoveries and focus on a steady flow of new candidates to move into the clinic. These platforms must be able to evolve over time. Life science has also become increasingly specialized, with each phase of the innovation cycle requiring specific skills and expertise. And finally, life science companies have to be able to collect and bring together data from a complex matrix of sources. New innovations in lab automation, gene editing, and genomics have intensified this complexity. Companies need ways to bring complex and connected data together to drive intelligent decisions. Unfortunately, the software to support this transformation has not kept pace with where the industry is headed. Our customers have shared with us the pain points of their existing solutions, whether it's paper-based, spreadsheets, homegrown solutions, or siloed point solutions. We hear from scientists how frustrating it can be to always be entering data manually into different systems, then spend time trying to piece it back together for review presentations. This not only wastes time, but it impedes the flow of innovation. R&D leaders also struggle to have the data they need to make timely and data-driven decisions. They have often become dependent on others to integrate and compile data from multiple sources, which can lead to delays in reporting or missing data sources. And the sheer number of applications used and the siloed nature of many applications makes it very costly and inefficient for IT teams to maintain. They often are using costly resources to maintain software instead of investing those resources in more impactful data science projects. Or they become dependent on their vendors for even simple configuration changes. It's clear that a new kind of digital solution is needed to help overcome these obstacles. We think every company and scientific team deserves a solution that will accelerate and support their innovations. When you think about the ideal traits that any digital solution should have in R&D, there are 4 attributes to consider. It has to be built for complex science by modeling the science you focus on, which means having both the data model built and fit for your purpose. It should adapt to your process, in that it has to support the processes and workflows that your team performs and have the ability to scale, iterate, and evolve over time. It should enable key decisions by empowering users with data at every level, giving scientists insights about their experiments and team leaders data on their programs, so you can make both scientific and operational decisions quickly with complete and accurate data. And finally, it should be easy and intuitive to use, ensuring that users will immediately recognize the value of the solution, see that it saves them time, and adopt it in a way that will help keep them compliant and consistent with the rest of the R&D organization. All of these attributes formed the inspiration for what we do at Benchling and drove the development of the Benchling Life Science R&D Cloud. The Benchling Life Sciences R&D Cloud is designed to give you a 360 view into your R&D innovation. Benchling's applications span the R&D process from experimental design, in silico biology, sample tracking, and process management, and our applications are all offered from a single interface. This makes it easy for users to leverage what they need without leaving the software. All of these applications are built on a common, unified platform. They share a configurable data model that maps to each team's unique scientific and operational requirements. This makes it possible to query the system for sample and process intelligence using our Insights application. Benchling is built on a modern cloud infrastructure, fully managed by Benchling, and offers state-of-the-art security and control. And we know that most companies have a rich ecosystem of instruments, software applications, and data stores that their R&D depends on. So we provide a full set of REST APIs and out-of-the-box connectors for lab automation, as well as ways to move data onto and out of the platform. Now let's look at a concrete use case in which the Benchling platform is being used to manage COVID-19 testing. You can't dedicate yourself to advancing life sciences without committing yourself to the ebb and flow of modern health. In light of the COVID-19 challenges, we launched the COVID-19 Testing Initiative to support those on the front lines of the epidemic. Testing workflows involve a number of logistical challenges, such as barcoding, sample tracking, and results reporting. Many facilities aren't set up to cope with these challenges at scale, and this can impact the speed of results reporting. These laboratories need software to streamline COVID testing operations. Now let's take a look at how Benchling could be used in your lab for testing processes and for handling large volumes of clinical data. What you see on the screen is a Benchling template. Using templates allows for standardization of protocols across teams and can help streamline lab processes. All of the steps outlined below can easily be tailored to your unique workflow. Now I'll be showing all of these steps in a single template to make things easy, but they can easily be split up into different segments, such as one template per lab station. The first step in this workflow is to log incoming patient samples. We support barcode scanning, spreadsheet import, or simple copy-paste. Now that we have logged the patient samples, we can track their physical location. In this example, we will be placing them into a rack. I can easily add the samples from the previous table, assign their location based on the barcode of the tube rack I will be placing them into, and allow Benchling to assign the next available location. Now that we have our samples prepared, we can get ready for RNA extraction. First, we will create a 96-well plate. Benchling supports scanning of pre-barcoded plates, or it can assign a barcode during plate creation. The next step will be to create and to plate our control samples. In this instance, I'm going to be using 3 controls, 1 positive and 2 negative. I will now put them into the plate that I just created. I'll choose my control samples here, assign them to my empty plate, and in this case, I'm going to manually choose the location for my controls as the last 3 wells in the plate. Now for the RNA extraction, we'll be using a liquid handler and Benchling's lab automation feature. This step can also be done manually if you're not using a liquid handler in your workflow. In this first automated step, or run, Benchling will generate an instruction file for the instrument, such as the plate barcodes. If the liquid handler produces an output file, for instance, one that maps the patient sample containers to the wells of the plate, that can be uploaded here. When I click Process, Benchling will automatically parse this output file and create a map of the resultant 96-well plate, showing the location of all patient samples, their newly created extracted RNA samples, and these new RNA samples will also be connected back to the patient sample from which they were derived. Now that we have our extracted RNA, we're ready for qPCR. In this run, we'll be mapping 4 96-well plates into 1 384-well plate. This again can be done through a liquid handler, or it can be done manually. When you're ready to record your qPCR data, if your analysis software needs a file mapping patient samples to each well in the plate, that can be done easily through another run. Using spreadsheet upload or manual entry, you can enter the obtained CT values from your qPCR run, designate the results as positive or negative, and load them directly into your template. Finally, The Benchling Insights application allows you to query data from our PostgreSQL warehouse. Dashboards like these can easily be configured to display number of positive versus negative samples tested, number of plates in each processing step, control performance, etc. Thank you for watching this presentation. Please contact us to learn how Benchling can be a solution to your research needs.